Hello, everybody. Good morning or good afternoon, depending on where you are. I'm happy to welcome you to this new seminar of the RNA Collaborative Network. And we have two speakers from the French RNA network, which is called the GDR RNA, and more precisely, RNA as a tool and a target for medicinal chemistry and chemical biology. So as you hear, this is a very interdisciplinary network. So the two presentations today will illustrate this interdisciplinarity, moving from chemistry until biology. And so the first speaker today is Mélanie Ethève Quelquejeu, professor at the Laboratory of Therapeutic Chemistry and Biochemistry, who will talk about RNA-cofactor conjugates as bisubstrates for the study of RNA methyltransferases. So Mélanie, I leave you the screen and I invite you to share your presentation.
Okay, thank you, Maria. Can you hear me?
Perfectly, yes.
Okay, so.
Yeah.
Okay.
Everything is fine.
Okay, so good morning or good afternoon, everyone, and thank you, Maria, for this kind introduction. I'm very pleased to be here today to share with you this project, which concerns the synthesis of RNA-cofactor conjugate as bisubstrate for the study of RNA methyltransferases. So first, I would like to introduce the team member. So here they are, the students, and I would like also to introduce you my two colleagues, Laura Yanazzo and Emmanuel Bro. So Laura is a CNRS researcher and Emmanuel assistant professor, and we really work together to develop different projects in our team. And we all are organic chemists, and our goal is really to synthesize molecules to explore and to interfere with biological processes. So as you can read on the title of our team, we are doing some heterocycle chemistry, such as in this context, some peptide chemistry, but most of our expertise is dedicated to nucleosides and nucleotides chemistry. And with this expertise in hand, we try to obtain some modification, some RNA, modified RNA. So first, I would like to show you why how we were beginning to be interested by this concept of bisubstrates. So we start our project on modified RNA by the development of chemoenzymatic strategy to synthesize stable amino acid tRNA. So this was quite long ago, and we tried to synthesize stable amino acid tRNA, since, as you all know, amino acid tRNA is a very unstable molecule. This is a tRNA which contains amino acid at the 3' terminal position of the ribose. And since there's this isomerization, it's a molecule which is very unstable in water, for example. So as a chemist, we decided to replace this ester bond by a very stable function, such as a triazole ring, an oxadiazole, or an amide bond. And we develop a strategy in which we are able to modify the size and the sequence of the RNA, the linkage here, and also the nature of the amino acids. So why we were interested to synthesize this kind of stable amino acid tRNA? This is due to the fact that we wanted to explore an enzyme that is involved in non-ribosomal peptide synthesis. This is this family of enzymes, the femtransferase enzymes, which catalyze this reaction. So as you can see, these enzymes use amino acid tRNA to added one amino acid to the growing peptidoglycan, and this is a bacterial enzyme which is involved in the synthesis of the cell wall of bacteria. So during this process, you will have the formation of an unacylated RNA, tRNA, and the addition of one amino acid to the growing peptidoglycan. So to study these enzymes, our colleagues which produce these enzymes need to have this stable amino acid tRNA. So with our molecules, we were able to develop the first inhibitor of these enzymes. We were able also to study the specificity of the enzymes by modifying, for example, the nature of the amino acids, and also the regiospecificity, which is involved in this mechanism. So this was the first steps, but then we wanted to go forward to try to obtain the crystallographic structure of the enzymes with this the tRNA part. And this is something which is usually quite difficult to obtain the crystallographic structure of the complex between the enzymes and the tRNA, the amino acid tRNA. So we tried with our stable analogs, but we didn't succeed to have some structural information. So that's why we go forward to this concept of bisubstrate analogs, which means that we want to develop molecules in which we could covalently link the two substrates of the enzymes. And then we were thinking that maybe we could have some very high affinity molecules for these enzymes, and we could have maybe the crystallographic structure of the complex. So this is what we did, and in this particular case, the bisubstrates, as you can imagine here, will be the covalently linked between the peptides and the molecules that will mimic the tRNA. So we synthesize this kind of molecule as which are in fact some peptidyl RNA conjugates, and for this we use these triazole links to connect these two molecules. And this was a good choice because these bisubstrates allow us to, so here are the different bisubstrates, the different peptidyl RNAs that we synthesize. As you can see, we can modify the RNA part. Here we choose microhelix to mimic the tRNA, is a very short part of the RNA. Here we modify the linkage. And with this molecule, for example, we were able to obtain the first crystallographic structure of the enzymes with this RNA part. And we were also able to explore the mechanism which is involved in the transfer of this amino acid. And as you can see, we obtained some very some molecule with very high strong affinity for the enzymes with an IC50 of 90 picomolar. So, and the last example I would like to show you by using this peptidyl RNA as a bisubstrate is this one. Quite recently, we developed a project in collaboration with Peter Levij and Dr. Mathieu Fonvielle, in which we were able to replace the RNA part with some xenonucleic acids. So, which means that we replace the nucleosides here by the HNA, the FANA, or the 2' fluoroRNA. And with each of these conjugates containing these different modified nucleosides, again, we were able to obtain the crystallographic structure of the complex, which allow us to look very carefully the conformation of these different xenonucleic acids. So I don't have time to talk about this project today, but you have the reference here if you are interested with this project. So all of these examples show you that it's quite interesting to use these bisubstrates analogs to study RNA-dependent enzymes. That's why we try to use this concept in to study RNA methyltransferase. So my talk will be divided in 3 parts. First, I will talk about the biological context, and then I will show you the synthesis of small molecules, which are the adenosine cofactor conjugates, and then the synthesis of RNA cofactor conjugates. So I'm sure that you all are aware about the importance of post-transcriptional modification in RNA, which is involved in many biological processes. And here we were interested by a specific modification, which is the methylation at the CEST position of the exocyclic amine of the adenosine in the RNA. So this modification was discovered more than 40 years ago in humans, bacteria, and virus. At the beginning, people were thinking that it was a static and unalterable modification, but in 2011, the demethylating protein, the FTO protein, has been discovered, which is associated with obesity. So this methylation, the M6R RNA methylation, could be found in tRNA, in messenger RNA, and in ribosomal RNA. And 3 families of enzymes are involved in this modification. First, the writers, which will methylate the RNA. The eraser, which will be involved in the demethylation, and the reader that could recognize specifically the methylation and will be involved in degradation, maturation of traduction. So to better understand this process, we focus, yes, and one important point is that the dysregulation of this kind of process are involved in the development of cancer, neurological disease, and it has been shown also that this is something important in the viral infection and also in some mechanism involved in bacterial resistance. So here we were interested to develop some chemical tools to study the writers proteins, so the methyltransferase proteins. So here is a mechanism which is catalyzed by this family of enzymes. And as you can see, the methyl is brought by the S-adenosyl L-methionine cofactor that I will call SAM. And during this process, you will have these transition states will be formed, which shows the SN2 mechanism during this process. And then you will have the formation of the M6A RNA and the release of the SRH. So there are not a lot of methyltransferases that are co-crystallized with the RNA substrates. As far as I know, there are just one example, the metal-16, which shows the crystallographic structure of the enzymes with the RNA partner. For example, for the metal-3, metal-14 complex, which is involved in the messenger RNA, in the human messenger RNA, they are just the crystallographic structure of the apoenzymes and with the SAM or the SRH molecules. So that's why that we were thinking that if we can develop some bisubstrate to study or to have some more information about the structural study of this methyltransferase, it will be fine. So this is a project that I developed with my colleague Emmanuel Bro, and it was a PhD thesis of Colette Abidjan. So to develop bisubstrate to study this methyltransferase, we want to mimic these transition states, which means that we want to covalently link a molecule that will mimic the cofactor here, attached at the N-cis position of an RNA part. We were not the first to think about bisubstrate to study methyltransferases, and there are some examples to study, for example, the nicotinamine methyltransferase. People try to synthesize this kind of molecule to have inhibitor, for example. This example shows you also the synthesis of inhibitor of protein methyltransferase or DNA methyltransferase. And when we began this project, there was just one example described in 1986, which was a molecule to inhibit the N7G methyltransferase of RNA. And now some of my colleagues in the south of France also developed some bisubstrates to study 2' O methyltransferase of RNA. So in our case, as I told you, we want to develop molecules to study M6A methyltransferase. And first, we start with molecules that will mimic just as an RNA substrate. We will just mimic the RNA with the adenosine. So now we have to develop the chemistry that will allow us to obtain this kind of conjugates. So this is what I will show you now. To mimic the SAM cofactor, we will use molecules in which we will replace the sulfur by the nitrogen, as you can see here. We want to keep the alpha amino side chain of the cofactor SAM, since it's very well known that there are very important interactions in the catalytical site of the methyltransferase. And we have to choose the good linker. So first, we choose to use an urea function as a linker here, or an alkyl linker. So today we will present the synthesis of these bisubstrates containing the alkyl linker. So to synthesize the analogs of the cofactor, we start from protected adenosine. We did an oxidation in 5' position to obtain this corresponding aldehyde, and then by using two reductive amination steps and one deprotection steps, we were able to obtain these molecules that will be the mimic of the SAM cofactor. As you can see, all of the functions are protected here, and we have just this amine, which is free, ready to react on the C6 or N6 position we'll see of the adenosine. So now we have to choose, as I told you, the good chemistry to be able to do the linked in this position. And for this, we use a strategy which was described by Lachman in 2007, which is the use of a convertible nucleoside. So we start from this protected inosine, which could be activated in situ by this reagent, the PBOP reagent, to obtain the benzohydroxyl activated inosine here. And then, since we put this compound as a reagent, you have this amine, which could react exactly at this position following nucleophilic aromatic substitution to give this conjugate with the adenosine here connected to the SAM mimics. So this reaction works well with some quite good yield of around 80% yield. So now we have to remove all of the protecting group, and this was quite difficult process, but we succeed in 2 steps to remove the terbutyl dimethylsilyl group by using this fluoroammonium reagent. And then by using this zinc bromide, we were able to remove the BOG group and the terbutyl ester here. So with this strategy, we synthesize a series of adenosine SAM cofactor conjugates as a bisubstrate. As you can see, we have some linker with 2 carbon, 3 carbon, without the side chain here, or with the alpha amino side chain, and 2 compounds containing the urea linkage. So this compound was used to study a model of methyltransferase by our colleagues from the IBPC, Institut de Biologie Physico-Chimie, Dr. Karine Tisney. And we choose these enzymes, which is a methyltransferase that methylates the ribosomal ribosome. This is a bacterial enzyme. And we choose these enzymes since it's a good model, since these enzymes the crystallographic structure of these enzymes was described in 2013. The APO enzymes, the crystallographic structure of the APO enzymes was described, and also the complex with the SAM and the SAH and the monophosphate adenosine. But no structure of these enzymes with the RNA was already described. So the team of Karine Tisney, they test and they try to use our molecules to do, and they do some assay, some DSF assay, and also some crystallographic structure assay. So they show that the best compound was these 2 compounds, which contain the alpha amino side chain. And they were able to obtain the crystallographic structure with this one, with a 3 carbon linker. And as you can see here, if we compare the structure that we obtain, so with our compound in blue, if we superimpose the structure with the structure obtained with the SAH, you can see that the alpha amino side chain is very well superimposed. But this is not the case for the nucleic base. So which means that this concept of bisubstrate is a good starting point, but we should optimize our molecules to maybe have to have something which is better positioning in the catalytical site of the enzymes. So to optimize this compound, we decided to enhance the size of the RNA substrates. So that's why we try now to use the chemistry that we developed for this conjugate to have access to some RNA SAM conjugates. So to introduce this modification, the connection of the cofactor into an RNA, we want to use the chemical strategy, which means that we need to obtain the modified nucleosides, and we need to synthesize the corresponding phosphoramidite modified nucleosides, which will allow us, by using the classical phosphoramidite chemistry, to obtain the corresponding RNA. So first, we have to synthesize this activated hydroxybenzotriazole inosine to be able to obtain the modified RNA. So this is what we did in 6 steps, starting from the inosine, and we were able to obtain the corresponding phosphoramidite. So now, my colleague, Dr. Laura Iannazzo, was able to synthesize, as I told you, by using the classical phosphoramidite chemistry. She synthesized the dinucleotides, the corresponding dinucleotide, and you can see that this hydroxybenzotriazole activated inosine is stable in this condition, or even the trinucleotides. And which shows the sequence, the AA, which is recognized by our model enzymes, the RLMG. And then the last steps is to see if this nucleophilic substitution is, the aromatic nucleophilic substitution is possible on the RNA part. And this was done by Laura Iannazzo also. And after the 2 deprotecting steps, we were able to obtain the some analogs cofactor linked to a small part of the RNA. So this is the molecules that we synthesize, the AA-SAM, the GA-SAM, and the cofactor in the intern position of the GAA. And to enhance again the size of these molecules, we use chemoenzymatic strategies. This is a strategy that we use to synthesize our stable amino acid tRNA that I show you at the beginning of my talk. And to use this kind of strategy, you need to synthesize the corresponding dinucleotides with your modification in 3' position, for example. This dinucleotide has to be phosphorylated in 5' position, and then it's possible by using the T4 RNA ligase to obtain the corresponding RNA. So we tried this ligation with this quite complicated dinucleotides. We were not sure that this ligation could work with this by the fact that we added all of this part of the molecules, but we were lucky to see that our dinucleotides phosphorylated in 5' position, even if you have this mimic of the cofactor linked to this position, the ligation works well, and we were able to obtain the 30-mer RNA-SAM cofactor conjugates. And again, we use the sequence here, which is recognized, which is the substrate of the RLMG enzymes. Okay, so now we have all of these molecules, and we were able, our colleagues were able to obtain the crystallographic structure with this compound, the dinucleotides connected with the SAM cofactor, with the crystallographic structure at 2.3 angstroms, and we were able to see the interaction between the enzymes and our conjugates. As you can see here, now the cofactor is very well, the compounds that mimic the cofactor is very well superimposed with the SRH. The A substrate is bound in the presumed substrate binding site, and the guanosine binds in the hydrophobic pockets. And with this structure, we could also propose a mechanism in which these 2 amino acids could play a role during to activate the amine during the transfer of the methyl group to produce the M6A RNA. So this is again an example in which this B substrate could help to show you that this B substrate could help to study this kind of enzymes. And currently, we initiate a collaboration with Professor Amedeo Calflich at the University of Zurich. And he tried to, they are using our molecules, and we have first results, which is quite very promising, because they were able to co-crystallize our compound with the metal-3 enzymes. And as you can see here, again, we have a very good superimposition with the structure that was described with the SAM cofactor. And just to finish, as you can imagine, the linker that we use with this to synthesize these bisubstrates could have an influence on the binding of the enzymes. So now we try to explore other kind of linkage. And as you can see here, we developed recently some bisubstrate in which we perform the links between the cofactor and the N6 position by using a triazole ring, which is chemically quite easy to perform by using the classical click chemistry, starting from an azidoadenosine and with an RNA which will contain a propargyl group at the N6 position. But as you can see here, we don't have the methionine, the alpha amino side chain. So to introduce this part of the molecules, we develop a new process in which we use this specific copper catalyzed cycloaddition by using an iodo-copper catalyzed cycloaddition, which is shown here. And by using this condition in presence of sodium iodine, it's possible to have this intermediate, the iodotriazole, which allow us, by doing some palladium cross-coupling, to connect this part of the molecules that will mimic the side chain of the cofactor cell. So this is an example to show you that this molecule is also a good way for the chemical point of view to develop many kind of methodology to access to more complicated molecules, or at least to have some molecules that will mimic better the substrate and the cofactor. Okay, so I hope that I convince you that this bisubstrate strategy could be useful to study this kind of RNA-dependent enzymes, and we already have done many studies on this RLMG model, and we hope to have nice results on the study of metal-3, metal-14. I would like to thank all of my collaborators that help us in all of these projects here, and my colleagues and the two PhD students, Dylan Coelho and Colette Adjian, which were involved in this project, and the funding was done by the ANR, the Agence Nationale pour la Recherche in France. And I thank you for your attention, and I'm ready for discussion or for question if you want.
Thank you very much for this excellent presentation, very interesting. So we have a few questions, but I would like to remind to the audience that you can ask your question using the Q&R window that you can access the bottom of your screen, open it and ask your question. So we have some questions from someone you may know, Jean-Jacques Pasteur, who is asking two questions. In the RNA peptides conjugates you described in the introduction of your talk, is the affinity of the conjugates of the FEM enzyme depending on the length of the RNA sequence?
Yes, sure. If you use, so when we try to, the longer RNA you will have, the better affinity you will get. But for the crystallographic structure, it's the inverse, which means that when we decrease the size of the RNA, we were able to obtain the crystallographic structure, because it should be very flexible when you have the full tRNA. But it's better to have a long size of RNA to have a good affinity of your compound.
Okay. And the second question is, does the affinity of the RNA SAM analog conjugate is better than the dinucleoside showed before? So the RNA oligonucleotides, I guess the answer is as previously. So the oligonucleotide has better affinity than the dinucleoside. The longer oligonucleotide.
You mean the adenosine SAM cofactor conjugate compared to the RNA conjugate?
Yes, I think so.
In fact, we don't know really because, as you can see in this project, we really focus on structural studies. But now we really want to have some access to some IC50 of the methyltransferase and to test this compound as inhibitor. And this is something that we are working on, but we don't have enough information to answer to this question.
Okay, I just have a last one. Which is the minimum affinity that you need in order to have the crystal structure in the end? Do you know? What is the affinity? I mean, you are in the low micromolar. Could we be in higher than that? Or
no, I think it's not really, we don't know. And sometimes you have some compound with very good affinity, but it's not very well placed in the catalytic site. For example, some of our conjugates are taking the bisubstrates, the two base are, you have a p stacking and you got the crystallographic structure, but it's really not relevant compared to what's happened in the biological media. So you don't have really a correlation between both.
Okay, thank you very much. We need to move to the second speaker now. Thank you again for your excellent presentation. And if someone has some questions, you still have time to ask Mélanie these questions in the QA window. And now we will move to the presentation of Zeynep Baharoglu from the Institut Pasteur in Paris, who is going to talk about epitranscriptomic adaptation to antibiotics, queuosine tRNA modification in Vibrio cholerae. And I leave the screen to Zeynep. Welcome and go ahead.
Thank you very much for the introduction, and I'm very happy to share our work with you today. So I will also talk about RNA modifications, but more in a biological point of view, and particularly in bacteria. So I will talk about epitranscriptomic adaptation to antibiotics in Vibrio cholerae, which is our model gram-negative bacterial organism. So we are working on the effect of low doses of antibiotics on bacterial stress responses. By low doses, I mean doses which do not kill bacteria, but still trigger stress. They are doses that can be found in the environment through release from industry, healthcare, and animal husbandry. And they can be released in the aquatic environment and soil, where various bacteria coexist. And we call them sub-mic antibiotics because they are sub-minimal inhibitory concentration. They are stressors for bacteria. And one question is, can they lead to tolerance or resistance acquisition? So just few words of definition of terms that I will use in this presentation. So resistance and tolerance are not the same thing. So antibiotic resistance is characterized by a higher MIC, which means that you need more antibiotics to kill a cell, bacterial cell. And thus it's resistant to this antibiotic. While tolerant population is not the same because it needs the same dose of antibiotic to be killed, but killing is slower. So if you plot the survival rate after antibiotic treatment, one population will die faster, but the tolerant one will take longer time to have the same killing effect. And this is also observable if we do competitions between two populations. If we have a tolerant red population and a non-tolerant blue population, we mix them in equal proportions. And then after several generations, if we count the number of red and blue, we will see that the red has won. So there is a more tolerant population remaining in the culture. So tolerance can lead to tolerance to effective treatments of infections, and also to relapsing infections where you treat bacteria. The infection is cleared, but then sometime after that, the infection begins again. So in order to look for factors that are involved in such tolerance in bacteria, in Vibrio cholerae, we did some transposon insertion sequencing, where we construct libraries of transposon insertions, which randomly insert in the genome in each gene. And then we collect these libraries and we grow them either without antibiotics or with the antibiotic of interest at doses below the MIC. And then we sequence the regions flanking the transposon. So here is an example of a gene that is important for growth. So before growth in antibiotics, you get these kind of sequences. So this is the number of sequences in each gene. And after growth with antibiotics, you see that the number of sequences in gene B has completely disappeared. It means that inactivation of gene B is lethal for that bacteria, then the bacteria disappears from the population. So we can identify important genes like gene B, and we can also identify genes for which inactivation is beneficial. And in this case, we would have enrichment of reads. So when we did that for several antibiotics. Here I'm showing a volcano plot, which shows the fold change of reads after transposon sequencing. So if the number of reads decreases, it means that these genes are important. And if the number of reads increases, it means that deletion of this gene is beneficial. And the y-axis shows a negative p-value. So when we looked at these plots, we saw a big number of RNA modification genes that came up. So some genes were beneficial and some genes were detrimental for growth in antibiotics. And we got tRNA modification genes and rRNA modification genes. So as you know, RNA modifications are derivatives of UCAG nucleosides. For tRNA, there are about 80 that have been described in bacteria and 35 on rRNA. But in bacteria, it's an understudied domain because the full modification landscape of tRNAs have been characterized only for 2 species, 1 gram negative E. coli and 1 gram positive Mycoplasma. And so the majority of these modifications is not essential for growth in lab conditions. And the deletion of these genes doesn't lead to any detectable physiological phenotype, again, in lab conditions. So people have studied these genes in a functional way, biochemically or for codon decoding, but no physiological phenotype has been described for many of them. However, they are highly conserved, so they are needed, they are conserved in the genome. And in red are indicated the ones that we have identified associated to the response to antibiotics. So we constructed 25 tRNA or RNA modification gene mutants, deletion mutants, and we did competition experiments, as I explained, to see with the wild type, to see if the wild type wins, whether the deletion mutant wins or whether nobody wins and there is no difference in fitness. And so this is, the graphs here show competitive index So it's in logarithmic scale. So 0 means there is no difference in fitness between the two strains. If it's positive, the mutant wins, and if it's negative, the mutant loses. And for most of the mutants in non-treated conditions, so here it's just growth in rich media, we confirmed the data in literature where there's no fitness defect of the mutants, where you can see that most of them are at 0, except a few of them. So there is no fitness defect in the absence of antibiotics. But when we when we do the same experiments in the presence of submic of antibiotics, we tested 4 different families of antibiotics targeting 4 different processes. We see that there are differences, and the differences are antibiotic specific and RNA modification specific. For example, for this one, TGT, it has a very strong 100-fold growth fitness defect in the presence of aminoglycosides, but not in ciprofloxacin or in trimethoprim. We have also some the beneficial ones. For example, when you delete USP here, its deletion is beneficial in the presence of aminoglycosides, but not with ciprofloxacin. And this was true for many of the genes, so we have specific responses. So we decided to further study this one, TGT, with a strong phenotype to understand what's happening. So TGT is making the queuosine modification on 4 tRNAs. It has been mostly studied in eukaryotes. So in mammalian cells, it's associated with oxidative stress response and cancer. In yeast, it's associated with mitochondrial protein expression. And in bacteria, there's no phenotype described for delta TGT mutant, physiologically. So we find a really strong phenotype here with antibiotics. So it's the first time that we see a strong phenotype for this mutant. So it has a fitness defect with aminoglycosides. And it also, when we test higher concentrations of antibiotics, so this time lethal concentrations, we can see that here it dies faster than the wild type strain. So it has it's involved in response to aminoglycosides, and as I said, the queuosine modification occurs in 4 tRNAs: tyrosine, aspartate, histidine, and asparagine. And for each of these 4 tRNAs, there are 2 codons decoded for only 1 tRNA, which can be modified by TGT or not. So we wondered whether all of these tRNAs are involved in this phenotype or only 1 or 2. So what we did is to construct some molecular reporters with the GFP fluorescent protein, where we introduced repeats of 6 times the codon we want to test. So this is a kind of a harsh reporter where we want to amplify the defect or advantage of decoding for the mutant. So here we have either 6 times TAC, 6 times TAT for each of the codons that we want to test. And the fluorescence will be a proxy for efficiency of decoding of these codons. And usually we saw no major changes for aspartate, histidine, and asparagine, but we saw a difference for tyrosine TAT codon. So this is a ratio of fluorescence in delta TGT over wild type. And here we see that the fluorescence is increased in the absence of the TGT for the TAT codon. So this suggested that there is more efficient amino acid incorporation at the tyrosine TAT codon in the absence of TGT and queuosine modification. So we also constructed another reporter, which is a gene called beta-lactamase. The beta-lactamase encodes beta-lactam resistance protein, which has a catalytic tyrosine. So this tyrosine is essential for its function, and it's a TAC or UAC in the mRNA. And we just did a synonymous mutation to UAU, which is also a tyrosine. So both versions code for the same protein, but it's not the same codon. And while in the wild type, both versions grow the same in the presence of beta-lactams. In the delta TGT strain, where all the tyrosines are unmodified, the version with the TAT grows faster than the versions with the TAC, meaning that it's more resistant to the beta-lactams. So this was consistent with the GFP reporter results, and it also shows that codon usage can affect how a given protein is translated by modified or unmodified tRNAs. Okay, so we show that absence of queuosine impacts tyrosine codon decoding, but we wanted to show the link between this and the actual phenotype of resistance or susceptibility to aminoglycosides. So we did some proteomics analysis. Here it's again a volcano plot showing protein abundance in delta TGT versus wild type in the presence of tobramycin. We see that many proteins are less abundant in delta TGT with tobramycin, as expected, but one was really highly abundant. There were 13 times more RS6A in TGT than the wild type. And we wondered if this would be involved in decreased viability in tobramycin. So RS6A briefly inactivates the response to superoxide stress or oxidative stress. And tobramycin is an oxidizing agent in Vibrio cholerae. So we hypothesized that in delta TGT, since there is more RS6A, maybe the oxidative stress response is less efficient. And we checked this by overexpressing RS6A in the wild type. And in fact, we can see here that when RS6A is overexpressed in presence of tobramycin, the growth is decreased. So high RS6A could be involved in decreased viability in submictal. So then since the protein abundance of RS6A is higher in delta TGT, we wondered whether this was because of more transcription of this transcript at the transcriptional regulation. But the mRNA levels of RS6A are unchanged in the absence of antibiotics and in the presence of antibiotics in TGT compared to wild type. So it's a post-transcriptional regulation. And what was interesting is that RS6A has a codon usage bias, biased codon usage towards TAT, which means that it has 6 tyrosine codons, and 5 of them are TAT, and the last one is TAC. And this is so it has 83% TATs, while in an average protein in the genome, it should be 53%. So half of them would be TAT and half would be TAC. So to ask whether the high levels of RS6A in delta TGT is because of its bias to TAT, we mutated all the tyrosine TAT codons to tyrosine TAC codons, so synonymous mutations. So the resulting protein is the same, but the codons are different. And we fused these two versions to GFP. And what we see is that the TAT version, the wild type version, in fact, is more abundant in TGT. But when we mutate all the TATs to TACs, we don't see this increase anymore. And we also tested this with the native GFP protein, which has 11 tyrosine codons So we made two versions, tyrosines which are only TAT and tyrosines which are only TAC. And here again, in TGT, in the delta TGT mutant, we have more fluorescence, and when the tyrosines are TAT, but not when the tyrosines are TAC. So this brings me to the concept of modification tunable transcripts, which were defined in eukaryotes first in this paper as transcripts which use specific codon biases, which encode stress response proteins, and for which translation is influenced by tRNA modifications. And we believe that RS6A could be classified as a MOT because it has a codon usage bias towards TAT. It regulates the SOX regulon, and its translation is influenced by queuosine modification. But in order for this to be a regulation mechanism, the queuosine modification levels should be dynamic, because otherwise it cannot regulate. So we asked whether there are conditions where queuosine modifications increase or decrease on tRNAs. And we found that, in fact, this is transcript levels of TGT, who first checked whether the TGT expression can be modulated. And we found that tobramycin increases TGT expression. And this is SHX is the stringent response. So the stringent response also induces TGT. And in a delta CRP strain, TGT expression is increased. So CRP, which is a major transcriptional regulator of bacteria, so CRP represses TGT. And then we wondered whether the expression of TGT is correlated with the actual modification levels on tRNAs. We first checked this using mass spectrometry. For tobramycin, we couldn't see anything because I think the difference is too low to be sensitive enough to detect in this technique. But in the delta CRP, we see increased queuosine modifications on tRNA. And then we did some Norder blots to detect queuosine modification. And this time we see that even with tobramycin, queuosine modifications increase in tRNA tyrosine. This is only tyrosine. So we propose that upon antibiotic stress, the TGT enzyme levels are increased, which leads to changes in RNA modification levels, more queuosine in tRNA tyrosine. And this impacts protein synthesis on specific proteins with TAT codon usage bias, tyrosine proteins. So this leads to translational reprogramming and adaptation to an adaptive response to the stress, which is in this case the response to oxidative stress and return to homeostasis. We propose that there's an epitranscriptomic adaptation to antibiotics, but not only to antibiotics, to stress in general, for example, to changing conditions. For E. coli, it could be gut versus urine, because it's the two niches of E. coli in the human body, or for Vibrio, it can be gut versus water. And we can think that the unusual codon usage for specific transcripts could be there could be a general mechanism of regulation through RNA modifications, because if you look at transcriptional regulators of stress, usually for some codons, they show strong biases, ribosomal proteins also, and also horizontally transferred genes, so exogenous DNA uptake, because by definition, this DNA comes from other species, so has different GC content and thus different codon usage maybe. And then we can also ask whether such transient tolerance due to translational reprogramming after RNA modifications can give the cells an increased chances to get genetic mutations and to become resistant. So with that, I am done. I will like to acknowledge the people who are involved in this work, especially Luna and Leo and Andre, who participated in this project, and our collaborators, Olivier Nami's group in IEBC Orsay, Yuri Motorin and Virginie Marchand, who help us detect modifications by sequencing, and platforms at Institut Pasteur, and Valérie Decrecy for discussions. Thank you very much.
Thank you. A very interesting presentation, very complex problems of bacteria, of course. So there is a question already, but then I will have, I would have many, so maybe at one point we will discuss together. But I will begin with the one on the QA, is Jean-Jacques Pasteur, who thank you for your talk. And from a structural point of view, do you know what is the effect of G replacement by Q in tRNAs?
Yes, so we don't know. So it's not trivial, because the G is the perfect Watson and Crick base pairing with the TAC codon when there's the G. But apparently, when the G is replaced with Q, the efficiency of decoding of this codon is increased. So, so the problem is complex, as you said, because we could expect that the perfect base pairing would increase the decoding efficiency, but it's not the case. So we don't know, yes, really.
Okay, thanks. I will have another question. So you study the epitranscriptomic adaptation in tolerance of bacteria, but there are many bacteria developed many mechanisms to tolerate antibiotics or stress conditions and persist. So I'm thinking, for example, about toxin antitoxin systems, and there are also others. Do you think these adaptation mechanisms are related, or they just go independently one another? And so if you tackle one, then you will have another tolerance.
Yeah, it's a very I think it's an important question. Maybe the timings could be different. Maybe so I think the post-transcriptional regulation is an additional layer of regulation to the transcriptional, which is well characterized, and we know it works. It could be a way of regulating essential genes, for example, because essential genes, you cannot silence them or mutate them like ribosomal proteins. But with this, only by RNA modifications and transient decrease of their translation, maybe it's easier to regulate this kind of genes. Okay. And maybe they are connected because, as I said, maybe if there is a post-transcriptional regulation of a transcriptional regulator, a stress regulator, then the stress regulator will transcriptionally regulate its regulants. So it could be there could be a connection like that.
Yeah, that's a very complex issue.
Yeah.
We have another question in the question and answer. So from the audience, from Marca Grai, who is asking, did you perform a proteomics to see if several proteins are up or down regulated in the TGT delta strain and check for codon bias?
Yes, we checked for codon bias in proteomics data, but we can't see any differences because I think the proteomics data includes many things, transcription, translation, but we did also ribosome profiling in delta TGT, and when we do that, we see that in ribosome profiling, the transcripts that are up are enriched in TAT, and the transcripts that are down are enriched in TAC. So it's consistent with the reporters.
Okay, a last question, and then I think our time is finished. When you checked the different antibiotics, it seems that aminoglycosides are the one with the most important effects on the mutants. Do you think this is related to the fact that these are RNA binders? Or
yeah,
yes, is not
actually we focused on aminoglycosides, but we had a very strong effects also with trimethoprim, for example, which impacts the DNTP pools. Mm-hmm. So it could be some correlation for some with the fact, especially for maybe ribosomal RNA modifications with aminoglycosides, which bind the ribosome, but it's not only that. Apparently.
Okay. Okay. Thank you very much. I would like
thank you very much
of you, Mélanie and Zeynep, for your presentations and the audience for staying and listening to the conference, and RNA Collaborative, of course, for having GDR RNA in the network. And the next seminar will be from University of North Carolina RNA Research Center on May 24th. So don't hesitate to register for this next seminar.
Thank you.
Bye, everybody. Thank you.
Bye.